morning, children. So this is the continuation part of the lesson story of microorganisms part 1. Yes, in the previous lesson we have done about the history of microorganisms, that what are microorganisms, that is who discovered them. So this we are introduced about it, and we recall for some of the part of this and again proceed the lesson to the lesson. Okay, children, so what are microorganisms? Microorganisms are the tiny microscopic organisms which we cannot see with our naked eye. It is through the advent, that is the invention of the cell, that is the discovery of the cell by the Robert Hooke in the year 1665 paved the way for the knowing of this microorganism into the world. So the knowledge of this science existed in the year 1674. In the year 1674 by Anthony van Leeuwenhoek. So this scientist has developed a lot of experiments through his single lens microscope and observed the various organisms through which he found out the first living creatures as the smallest living creatures as bacteria. So he was the first scientist who observed in 1674, and it paved the way for the microbiology. So in 1674, the study of microbiology came into existence. So several scientists worked out about the microorganisms and found out that there are very minute microscopic structures and the organisms in this world which cannot be seen with the naked eye. They are invisible. They say there is also an invisible world through which we cannot see with the naked eye, but which can be seen only with the help of the powerful microscopes. So what are these microorganisms? How are being they explained? I'll show you now that there are different types of microorganisms. I have mentioned you, like bacteria, algae, fungi, protozoans, and microarthropods. So these are all microorganisms are omnipresent. I said you before that they are omnipresent. They are present everywhere. That is in water, on land, in air, in human beings, inside the human beings, on the human beings, everywhere. They are present everywhere. That's they are called as omnipresent. So they survived from the ice cold climate to hot climates. Marshy land, they are present in the deserts and as well as in the marshy lands. So they are present in all the temperature region, ice cold climates to hot climatic conditions. Did you understand, children? So they, under all types of environmental conditions, they can survive. The hot springs, hot climatic conditions, deserts, they are existed in the deserts and also marshy lands. So they can survive under all the environmental conditions, we can say. So the microorganisms can survive under all the environmental conditions, and they are omnipresent, present everywhere: air, water, land, food, all the human beings, inside the human beings, in the animals, everywhere, in all the animals, they are present. And the different climatic conditions also, they vary. At different environmental conditions they can survive. And we can see that there are different types of microorganisms, especially one by one. We'll see about the bacteria first. They are the first microorganisms observed by the scientist Anthony van Leeuwenhoek. So types of bacteria we'll see now. What are the different types of bacteria? Okay, children, bacteria are of different types. They may be spherical like cocci, which are spherical in shape. Okay, bacillus, that is rod-like. They are in the form of rod shape. They are rod-like, and some are spiral, that is the spiraling. So they are in the form spirally arranged to one another. And the other one is comma shaped Vibrio bacteria. So they form the comma shaped. So this is the shapes of the different, they get their names, that is the cocci, bacillus, spirillum. So through the shape they occur, they get in. So they got these names, cocci, bacillus, spirillum and Vibrio. Next, we move to the, what are the cocci? So the bacteria, all the microorganisms, generally they occur singly, that is alone. They may be alone or they may be in colonies. They may be alone or they may be in colonies, or individually they may move freely. Sometimes move freely. Is it clear? So if it is one example, cocci if you take, they may be alone as such or they may be grouped as colonies to one another. This is called as the arrangement of they are arranged in the colonies. So they may be grouped as alonely, that is they move freely, or they may be in the form of colonies too. So what are the different factors? So different bacteria that are in the nature, we'll see with the examples. Note down first. Children, this is the structure of the bacteria, Bacillus bacteria. See here, the structure shows that it is a rod shaped and it contains villi and the flagella, which helps for the motility, that is the movement of the bacteria. It is minute and microscopic and it's very smallest one, and it contains the cell body. It's covered by the plasma membrane here, and this is a nucleus is not defined properly. It is not true nucleus, where generally the nucleus of a cell contains inside the nucleoplasm. The nucleus can be defined, but here in the bacteria it is not well defined. The nucleus here, they contain the cytoplasmic fluid, and these are the blue colored structures. These are the ribosomes which help for the protein synthesis of the cell. Next, the size of the bacteria varies to 0.2 microns to 100 microns, and now they are aerobic. Some they respire in the presence of oxygen, and some they also respire in absence of oxygen. Now they are anaerobic. They respire in absence of oxygen. So they require oxygen for respiration, some of the bacteria, and some of the bacteria can also survive in the absence of oxygen. So here we can see that the range of the bacteria from 0.2 microns to 100 microns. So one of the biggest bacteria, that is 0.75 mm, that is Theomargarita namibiensis, which is found in the coast of Namibia, which can be seen, that it can be seen through our naked eye. That is directly we can see this bacteria. Normally you know that these unicellular organisms cannot be seen with our naked eyes, but this Theomargarita namibiensis of the coast, which is found in the coast of Namibia, can be seen with the naked eye. Next we'll see how to culture. So how to see this bacteria? They are minute and microscopic, I said. So how to culture it? So we'll see in milk. You said that the milk contains the lactic acid bacteria, that is lactobacillus. Milk contains lactobacillus bacteria. Is it clear? So it helps, milk is converted to, milk, due to the presence of the lactic acid bacteria in the atmosphere, lactic acid bacteria, that is the lactobacillus in the atmosphere, lactobacillus bacteria, presence of this in the atmosphere, it converts the milk into curd. Milk is converted into curd due to the presence of lactic acid bacteria in the atmosphere. It helps the milk to change into the curd. So if you take the curd, it contains this bacillus, that is this bacteria can be observed in the curd. You take the liquid, that is a buttermilk, 2 drops of buttermilk you take and you put it over the slide, glass slide, and heat it from below. Is it clear? So, when you heat the slide from below, after 3 to 4 minutes, you wash the slide from backside and then you observe it under the microscope by staining it with a methylene blue. After that, you again wash it and then first you take a slide, put 2 to 3 drops of buttermilk over it and cover it with a cover slip, add the methylene blue and again after adding the buttermilk, you heat it from the down and add the methyl orange or methylene blue and cover it with a cover slip, wash it from the back side and then observe under the microscope. Once again, I'll tell the steps. First step, 2 to 3 drops of buttermilk on glass slide is to be taken. Next, and it should be spread. It should be spread. You have to tilt the glass slide in such a manner that it spreads over the glass slide. Then what to do? Then heat the slide and for 3 to 4 seconds let it be there. Heat it. Heat the slide. Second step is you have to heat the slide after adding 2 to 3 drops of buttermilk over the glass slide. Then add crystal violet or any other indicator. Crystal violet. Okay, children. Then add crystal violet. Wash the slide. Then leave it for 30 to 60 seconds. 1 minute rather. Leave it aside. Leave it for 30 to 60 seconds aside. Is it clear, children? Then wash the slide. Wash the slide with water. Wash the slide with water. First, add the 2 to 3 drops of buttermilk over the glass slide. Then heat the slide from bottom and add the crystal violet over it and leave it for 30 seconds or to 16 seconds. Then you wash the slide with water. Then observe it under the microscope. Observe the slide, keep it over the stage of the microscope and observe it under microscope. You have to follow these steps to find the bacillus bacteria. Bacillus is rod shaped, isn't it? So you can find the rod shaped bacteria when you observe the slide. You find the cells where you find the rod shaped bacteria completely. Is it clear, children? So this type you might be seeing under the microscope. So this is about the lactic acid bacteria, how to observe the bacteria through the experiment. You have taken the buttermilk and observed the bacteria. Now we'll see about the bacteria. So what are the different functions that the uses of bacteria? Uses of bacteria. We'll see now. Okay, children? See, children, uses of bacteria. You know that bacteria are present everywhere. So they are omnipresent. They are present on our body, in our body, and also they cause the diseases, you know, as well. But they are useful to us as they are present in the intestine. They help for digestion of the food. So present in the intestine in human beings and animals also, like horse and cattle, they digest the cellulose in cattle, that is the plants. The cellulose in the plants is being digested, that is being eaten up by the animals. So present in the intestine and help for digestion. So we can say that is also helpful in such reasons, and they are harmful as they grow on our skin and causes so many skin diseases too, as well as they cause so many diseases like typhoid, tuberculosis, septicemia, that is called as blood poisoning. Okay, children, next we'll move to algae. Okay, write down. Children, about algae now. So the algae, they are the simple non-flowering plants. They are the non-flowering plants, aquatic plants, they are found in water mainly. When the water is stagnant for so many days, you can find the formation of algae. These plants can be seen. Mostly they are the seaweeds, we can say. They are single-celled microscopic organisms, and they contain chlorophyll and they prepare the food by themselves. In fact, they do not contain the true stems, leaves or roots. They gather in the numerous forms, that is in the clustered form. They will be available in the ponds or in the aquatic regions. So for the example, chlamydomonas and spirogyra, the name itself tells us these are the flagella which help for the motility or the movement, and this is the spirogyra, it's in spiral shape. So this is the algae which are found in the ponds. Okay children, so these are the green scrapings of the side of the pond. If you take it and observe under the microscope, so you take it with a needle and put it on the glass slide and observe by adding the methyl orange or crystal violet and putting it cover slip over it and observe under the microscope, you can easily observe this algae. We say the pachi in Telugu, isn't it? That's can easily observe the algae under the microscope. Next we'll move to the fungi. Note down. Next group, fungi. Fungi, they belong to cryptogams, non-flowering plants, and they appear to be plants, but they are not plants. Their examples are mushrooms. They do not have roots or leaves. These mushrooms are unicellular and simple non-green plants. They cannot prepare their own food, and they are non-photosynthetic. That is, they depend on the dead, decayed organic matter for their food. So they are the example, as you know, that there is mushrooms, and they are heterotrophic. The name itself, since they do not prepare their own food, they are heterotrophic in nature. And these fungi, we can see over the bread, molds that are formed over the bread. So the picture, you can identify the spores, and these are the hyphae that produce over them. So that you can learn in the higher classes what are the hyphae and all sporangiospores, spores that formed over, and everything you can learn in the next chapters. Next, aspergillus is one of the examples for this fungi group of microorganisms. So these are also minute, microscopic, and can be observed only through the microscope. So molds can be observed, the mold that are formed on the bread can be observed by taking through the needle and putting over the glass slide. You can also observe the features of in this form under the microscope. Okay children, next we'll move to the protozoans. We have completed bacteria, algae and fungi. Now we'll move to protozoans. Note down. Next, coming to the protozoans. They are unicellular aquatic organisms. They exist alone or forms in the colonies also. They are free living, or sometimes they may be parasitic too. So they are irregular in body shape. Some are oval in shape or spherical, or sometimes they forms the different shapes. So they contain the single nucleus, and they are unicellular, and the single cell performs all the life activities in these protozoans. And you can see the examples as amoeba, paramecium. See here the amoeba, the structure and the cell formation, the cell membrane, the nucleus, cytoplasm and cytoplasmic fluid inside, and the food vacuoles inside. And next, this is the paramecium. It's in the form of slipper. So it's called as slipper animalcule. It's called as slipper animalcule. It's also a protozoan parasite. So hope you have understood, and now we'll see how to culture these protozoans and observe them. So how to culture these protozoans? For culturing the protozoan, you have to take the straw. Do you understand, children? So you have to culturing of the protozoans. You want pond culturing of, to culture the protozoa. The materials required are pond water, hay, tray. You need a tray to culture it. Okay children, so this hay you have to keep in this tray and put the water, pond water inside it. Okay ma? Next, you have to soak it for 4 to 5 days. You have to soak the hay for 4 to 5 days. Then it will be decomposed. The pond water that contains the protozoans, they decompose the hay, eat up. They take the food the hay as its food and decompose it, and they develop in more in number. They become protozoans becomes more in number. So when you take this 2 to 3 drops of this water after 4 days and observe under the microscope by putting it on the glass slide and and adding the crystal violet and covering with the cover slip and observing under the microscope, you can see the protozoan structures like this, irregular structures you can see under the microscope. Is it clear, children, how to culture the protozoa? That's the experimental process to culture the protozoa. Next, we'll see about the viruses. Now, viruses. They are the smallest microorganisms. They cannot be seen with the naked eye directly. Then the simple microscope also does not help to see it. It needs a powerful electron microscope to observe. See here, they are living when present inside the living cell, but they are not living when they are outside the living cell. They are being as a dead cells. So they are the heterotrophs. They cannot prepare their own food. They can survive only under the living conditions of the metabolic activities which takes place inside the living organisms. So you know that the present COVID virus is also a virus which survives only in the living organism in our body itself. So we are protecting ourselves by not entering the virus into our body through eyes, nose, mouth, etc. So they are taking the precautions accordingly. You know about this viruses now. So these are examples of poliovirus, TMV, that is tobacco mosaic virus. Normally it is attached to the plants. So note down, children, about the virus. Next, children, about the microarthropods, the animals with the jointed legs like the insects. The name itself tells arthropoda